I mean, what if I dress up like at the same
Yeah.
Why are you not working now?
I just have it right.
Yeah. Let's try this. Try it again.
Oh yeah, that's
How
my
do you do that? You push the pause button. Oh,
Oh what? Oh,
I
that's
pressed
my
pause on the timer. I guess that affects the advancing. Evidently not.
You guys,
Yeah,
so
who knows?
it's just a fun pairings that I have.
Okay.
But I don't know.
Okay.
Started. Fortunately, you closed the door. Hey, I'll introduce myself for those of you who don't know me. I'm Cassie, PhD, well, not yet, almost PhD student in Mitch Wiegman's lab. I'm one of the WildEcol coordinators along with Hannah, Portia, and Julia. We're always looking for more members to join, so if you want to help out, you're totally willing and able to do that. I'm just gonna start right now with just an acknowledgement that we're here in Saskatchewan on Treaty 6 territory in the homeland of the Métis. We pay our respect to the First Nations and Métis ancestors of this place and reaffirm our relationship with one another. So with that being said, I'm gonna invite Mitch up, who's going to introduce our speaker, Kayla. So everything should be good to go.
Sounds good. Thanks, Cassidy.
Welcome, everybody, to WildEcol. It's a real pleasure today to introduce Dr. Kayla Davis as today's speaker. Kayla comes to us from Virginia, where she lives with her husband Kyle in Richmond. And she's been in our group for less than a year, 8-9 months now, I guess already.
Yeah.
February-ish.
February-ish, so 9 months. Yeah.
Yeah, and Kayla's work leverages an immense background in quantitative ecology and avian ecology. Lots of experience with shorebirds previously, now with Arctic nesting geese. She has experience in the mystical ecology side of our discipline, and that's been really helpful for this particular project, where she's combining survival and reproductive success information for a variety of lesser snow goose colonies across our Canadian North. Kayla earned her undergraduate degree at Wake Forest University and a master's degree from Virginia Tech. She then went to Michigan State University to work with Elise Sipkin for her PhD. And with that, I think I'll stop on an introduction. It's been such a pleasure working together, and I'm really happy you could make a trip back. So welcome back to Saskatoon, and looking forward to your seminar.
Thanks, Mitch. Yep. Thank you for the introduction, and thank you for having me. Thanks for the invitation to present to your WildEcol seminar. So as Mitch said, I have worked in a number of different avian systems, and today I'm going to talk about one of the common tools or methods that I use a lot called integrated population models, and talk about some of the questions that I have addressed with this method. Let me start this timer. Okay, great. Okay, that works. So as ecologists, we are primarily interested in understanding how many things there are. What is the status of these things on the landscape, these animals, wildlife, and what is their trend? We might be interested in understanding a little bit more than that, though, right? Perhaps we're interested in understanding what affects that status and trends of these individuals. So here we have a relatively simple model where we're looking at the counts of individuals, and okay, I can't point to anything, but counts of individuals at some place at a certain time, the mean, and how that's affected by some environmental variables like the North Atlantic Oscillation or NDVI. So this can give us some information about how the environment affects these trends that we're seeing. We might also be interested in understanding how different demographic rates contribute to population growth. So here we're looking at how survival might contribute to population growth rate or population change. In this case, there's a positive relationship. So we would say that survival is a strong contributor to the change that we're seeing in population growth. But oftentimes, our data over a long time series can look something like this. So we can have multiple different data sets. Here we have population count data, productivity, and capture-recapture data over a span of 20 years. But the data can be sparse. It can have gaps in it. And if we look at each of these individual data sets as independent data sets, well, we can only say something for productivity from year 7 to year 15 here. For capture-recapture or survival, from year 11 on to 20. So we are kind of limited in the amount of time that we can make inferences over if our data don't cover our full time series or if they're not complete over that time series of our study. So the question I ask a lot with my work is, how do we utilize the data that we have available? We have all these long-term monitoring projects with long-term data sets that exist. And how do we use that to assess species trends and demographic rates in order to inform management? So that's a question that's at the heart of a lot of what I have done. So as we've talked about, I work a lot on demographic modeling of these species here of shorebirds, waterbirds, and most recently waterfowl. And these are pictures of some of the different species that I have worked with. These species are similar in some of the threats that they face. So many of them have long distance migrations and they face unique challenges throughout the entirety of the annual cycle. Many of them are dependent on coastlines or wetlands, and they face habitat loss in many of those systems. This figure here on the left-hand side of the screen shows a portion of a figure from the Rosenberg et al. Paper. This is the 3 billion birds lost paper, if you're familiar with that. And what they did was look at species declines as numbers of species or individuals lost since 1970. And for shorebirds and waterbirds in particular, they have declined a lot since 1970, but waterfowl has increased. So these are different species inhabiting similar landscapes, but seeing very different trends over time. So this brings up a lot of interesting questions for me personally, as someone interested in these species. So for the rest of the talk, I'm going to talk a lot about the method that I have used a lot recently called integrated population models. I'm going to start with a background on what IPMs are and how they're used. And then I have 3 examples here of how I have used them in my work to address different questions. So the first example is looking at population viability of a declining species, extending this model to multiple species, and then extending it to multiple populations. So first, a little bit of background. So integrated population models have become increasingly popular. They have many benefits that researchers can take advantage of. The figure here shows the count of IPM papers in the literature since 2001. And as you can see, over the last decade or so, those IPM-focused papers have really increased. And that's because there are a lot of benefits to them. So they combine multiple data types into a single unified analysis to study the dynamics of animal populations. We even have a textbook now dedicated to learning how to use these models, which I find extremely helpful. If you are interested in this, this is a really nice text. So as I said, IPMs leverage multiple data types in this single model unified analysis. So we can combine things like productivity data to look at fecundity or productivity, mark recapture data to look at survival. And when we link these to population size data, we can gain a mechanistic understanding of how different demographic rates are contributing to population change. So that's a real benefit of this model framework. This is a schematic diagram of what an IPM kind of looks like. To walk you through it here, the data are shown in purple diamonds. the biological process parameters, which are the demographics that we're interested in estimating, are shown with different color rectangles, and then the observation process parameters are shown with the red circles. So essentially what this is a large model composed of multiple submodels. Often we have some model to estimate reproduction. In this case, I'm using the chicks and the number of nests to estimate fecundity, which is linked to the number of newly recruited individuals in the population. You can also use banding data. In this case, we're using it in a simple CJS model or Cormack-Jolly-Sieber model to estimate adult and juvenile survival. And then these parameters are linked to a population model, which then estimates how many individuals there are. What is our population size? So that's the IPM background. First example that we have here is looking at population viability of a declining species under different management scenarios. So this work focused on black terns as our management case study. So this is a common species throughout North America. However, they have declined severely across their range. So this map here shows the annual declines in each of the states and provinces from BBS data ranging from 1970 to 2019. And this is the decline at the study site where these data come from. So you can see this is a really steep decline over the time series. So these birds are declining despite ongoing management intended to mitigate these declines. So we want to understand what's going on with these birds. So these birds are managed heavily on their breeding grounds, and like many other migratory species, black terns are managed at their breeding sites. And that's often for very practical reasons. So these birds are long distance migrants, right? And they're stationary during this breeding period, and they're often easier to access during that time as opposed to wherever they are in their non-breeding season. So some of the management interventions that I have featured here are these nesting platforms to increase nest success of black terns, area restrictions for the benefit of shorebirds, and predator control for in this case, I think it was for shorebird species. All of these interventions work to increase productivity, and they can be very successful at that. However, these interventions don't always translate to the population level increases that we would hope to see. And management to increase a population size should really focus on demographic rates that are going to lead to population change. And that's something that we can analyze in the IPM framework. So here our objectives were to estimate those demographic rates and environmental drivers of those rates for black terns. And then within a population viability analysis, project the population under realistic future climate and differing management strategies. Our survey data came from this breeding colony here in Michigan at St. Clair Flats on Lake St. Clair. The data were collected over a 10-year study period. The data consisted of adult count data for the population size, fledgling count data to estimate productivity, and mark recapture data to estimate the survival of adults and juveniles. And one thing that it's important to point out here, these black triangles are the historic locations of breeding colonies for these birds, and very few of those still exist. And St. Clair Flats is one of the largest remaining breeding sites for these birds, or known breeding sites for these birds. So this is a really important location. I'm not going to run through this IPM. Again, this is the same IPM that I introduced in the introduction. So this is the framework for this model. So we're estimating fecundity, survival, and population size. We were also interested in looking at how different environmental variables affected these different demographic rates, particularly fecundity and adult survival. So we used local scale climate variables to assess the effect of different variables on fecundity. These are things that we thought would affect nest success, chick survival through the breeding period. And then we also included broad scale climate variables, which were climate indices summarized over the wintering period. So this was the Southern Oscillation Index and the North Atlantic Oscillation Index, because we wanted to incorporate some sort of information from the non-breeding period onto adult survival. We found that the previous winter NAO had a positive relationship with adult survival, and this climate variable was used in our population viability analysis to project the population forward under different climate scenarios. I'm not going to go into too much detail about the overall results here, but this is an example of what you can expect to see from an IPM. This is your basic information, your population size estimates. I'm showing the observed counts in black and the estimate in their 95% credible intervals in gray and gray shading. Fecundity, which in this case is the number of fledglings per female, juvenile survival, and adult survival probabilities. In these demographic rates, the lines and shading are the mean of those rates, and then the dots or squares are the annual estimates, except for fecundity, that was the observed fledgling. We estimated mean fecundity in that case. So fecundity was really low. Juvenile survival was really, really low, but this was survival from fledging to age 2. So this is survival over, a number of years, so we expect this to be pretty low. And then adult survival probability is around what we see for other tern species, but slightly low. So moving into the population viability analysis, we used that relationship with the NAO and adult survival to look at the 100-year mean of NAO. We also projected with the 10-year historic mean of the NAO to look at 2 different climate scenarios, but I'm only going to show one today. And then these different management scenarios are looking at if we targeted different demographic rates and were successful in increasing them, what would we be able to see under kind of the best case scenario if we saw success? So we actually manipulated the actual rates estimated in the IPM, because we didn't have any covariate effects that would serve as good proxies to manipulate for changes in these rates. But that is something you could do. If you had, for example, I've seen an IPM with this population viability analysis where adult survival and chick survival was affected by the presence of Merlin falcons. And the scenario there was looking at the control of Merlins. If you reduce the population by a certain amount, what was the expected change in adult survival and chick survival that you would expect to see? Here, we just manipulated the actual rates. And those scenarios were to double the historic mean fecundity, which was really low. So we thought that it was reasonable to be able to double that. Increasing juvenile survival by 10%, increasing adult survival by 5%, a combined approach of all of the above, and then a no additional management scenario on top of what's already being completed. So there is quite a bit of management already being completed at this site. So that scenario is nothing on top of what's already being done. So we projected the population size 10 years into the future under these different scenarios, and we evaluated local quasi-extinction probability. And that quasi-extinction probability is something that we were really interested in, because if you'll recall from the map previously with those black triangles that showed the historic colony sites, many of those no longer exist. So this quasi-extinction probability is actually something that we're really interested in, because this is an important breeding site for these birds. So for our quasi-extinction results, the quasi-extinction probability is on the y-axis in this figure, and the projection year is on the x-axis. The different management strategies are different colored lines here. Under all the scenarios, population size decreased and quasi-extinction probability increased across all scenarios. The no additional management scenario produced the highest quasi-extinction probability, and the scenarios that included adult survival manipulation or management were the strategies that produced the smallest quasi-extinction probabilities. And I didn't show the population growth and demographic rate correlations here, but adult survival was the strongest correlation with population growth. So we would expect to see that management of this rate would have the greatest benefit for the population. Okay, for our first example conclusions, just for the demographic rate summaries, fecundity and juvenile survival were both low, and adult survival was relatively variable for what we expect to see. Current management efforts are unlikely to reverse the ongoing decline of black terns at St. Clair Flats, but those interventions targeting adult survival are the most likely to have fruitful effects for improving population growth. So that really points to the need to enhance monitoring on the non-breeding grounds and potentially enact conservation strategies and management there. And this approach is easily adaptable for other species to look at other situations where you might have different management strategies that you're interested in evaluating. This is a really good approach for evaluating those choices. The next example is this beyond single species, extending the model to multiple species. This is work that I did on an Arctic breeding shorebird community. So we're estimating species and community level rates with this extension. So as I said, we're working here with an Arctic breeding shorebird data set. The data were collected over an 18-year period from 2005 to 2022. The data come from a long-term monitoring project around Utqiagvik, Alaska. The data consisted of population counts, which were nest surveys, productivity data, which were the count of chicks hatched per year, and the survival data come from the adult banding, recapture, and resighting data sets. And these are the species that were a part of the community. So American golden plover, dunlin, long-billed dowitcher, pectoral sandpiper, red phalarope, red-necked phalarope, semi-palmated sandpiper, and western sandpiper. So our objectives here, one of our objectives was, is this a useful approach to do this? So we wanted to estimate and compare demographic rates between the species and community levels and determine the utility of this multi-species IPM or MIPM for estimating the rates and trends of some data limited species. So there were some species in our data set here that have very, very low site fidelity, and we had really limited survival data for them. And we wanted to know if this was a useful approach for estimating the demographic rates for those birds. In this case, the model looks a little bit different. We estimated adult survival only, so that was one difference in the banding data side. But here we estimated fecundity outside of the IPM, and that was because very few birds that were banded at this site returned to breed there. So we didn't want to incorporate fecundity into the population model directly. So we used fecundity actually as a covariate on this adults gained or immigration parameter. And the reason why we did that was because we had this idea that even though birds that were banded at this site rarely return, we thought that fecundity at the study site probably reflected fecundity at the greater land regional scale where birds could be recruited from. So birds might not recruit directly from the place where they were hatched, but they might be recruiting from the larger area. So we wanted to look at the effect that fecundity had on the number of new adults gained in subsequent seasons. So we had this additional parameter here. This is something that actually was not directly informed by data. This is a benefit of IPMs. You can estimate immigration, which is essentially what this is, and without any information directly informing the estimation of that. Now, there are some limitations to that if you really get into it, but that is another benefit about using this modeling framework. For the community aspect of this, you can really think about this as the community being a distribution of distributions. So here we have the individual species responses, which let's think about survival rates. So each species has some survival mean. So that mean would be the peak of this distribution. And then there's some variance around that mean within which all individuals would fall. So for each species in the community, they've got some survival mean. So this could be semi-palmated sandpipers, American golden plovers, western sandpipers, etc. But all of these individual species responses could be characterized as a part of this overall community response. So you've got this overall community mean that describes the individual species responses. And the width of that would be the variation that you see within your community. One change here that we've made to reflect this multi-species context is adding this index for species S. So now we're estimating all these different parameters for each species within our community. And we have our species level responses drawn now from this community level mean. And so now we can do that. This is actually quite a simple extension. We can do that for all of the species in our community. Or sorry, all of the parameters that we're estimating in the model. So we can get the same information, but now from one model, we get information for all of the species within the community. Here are the population size estimates for all of these different species. I'm not going to talk through all of these, but see a lot of them have kind of different things going on, but similar peaks and valleys over the time series for some of these birds. But the thing that I think is the coolest part of this is having that community level inference as well as the species level inference. So here are community level annual means for adult survival. So the black line here is the mean, and then the gray is the credible interval around that community level mean. So this is the mean survival for the entire Arctic breeding shorebird community at Utqiagvik, Alaska. For Dunlin, which is a species we have a lot of data for, you can see that in most cases, the species level response is pretty close to that community level mean. But in some years, this deviates. And we want to see that. We want to be able to see that there is some deviation for species from this community level mean, because if it's not that informative for us if we are just saying the individual species are equal to the community mean. That doesn't give us that much information, which is the case for a species like red-necked phalarope, which has maybe 3 or 4 recaptures ever. And so what we see here is this species level mean is drawn towards that community mean, and there's a lot of variation around that, a lot of uncertainty. And so basically, we can't say a whole lot there. In this case, we're at the limit of what information we can get from these data for this species. And that honestly is the case for many of the species that we see in this community. That many of them are data limited, and a lot of them are drawn to that community level mean, which may be truth, but we're working on simulations now to understand what are the limitations of this kind of modeling approach. How many species can be data limited in your data set? What if you have 1 or 2 species with very good data, and then the rest have okay data? How much are these individual species influencing the community mean, and vice versa? How much is the community mean influencing the other species within the community that don't have a lot of data? And last thing that we have done here, because we have these rates estimated for these different species, we can look at comparisons of pairwise species comparisons. Here on the y-axis, well, these are different pairwise species comparisons between American golden plover, dunlin, and semi-palmated sandpiper, and each panel is a different combination of species for adults gained and fecundity, which are kind of the breeding season demographic rates. And what I'm showing here is that these are pretty correlated, and that reflects some demographic, or what we think is some demographic synchrony. So these birds are likely responding to this, well, they are responding to the same environmental conditions, and it looks like they're responding in similar ways. So there is synchrony in breeding related demographic rates for these species. And interestingly, there was not synchrony. So there were no correlations, or the adult survival rates were not correlated among species. And we thought that was pretty interesting because these species have different overwintering ranges or non-breeding season ranges, and they use different migratory flyways, and they're experiencing different risks throughout the non-breeding period. And so we think, this could be an indication, a potential indication that these birds are responding to conditions during the non-breeding season, and that's affecting their survival more so than the conditions on the breeding season during the breeding season, which are similar, and that we would expect to see more synchronicity in their comparisons, in their rates. So to wrap up this example, while we were able to estimate those demographic rates for both species and community levels, some of the inferences that we could gain from those data limited species were maybe just okay. We've got to do a little bit more work to determine how useful this approach actually is. But one interesting thing that you could do to take these data or estimates further, or to look at the demographic synchrony and asynchrony, which could potentially point to limiting periods during the annual cycle. And finally, the work that I'm doing here is looking at assessing midcontinent snow goose metapopulation dynamics within this IPM framework. And I'm still working on this model a lot, and so I don't have super polished results to show you, but I think it's useful to show what I'm working on here, and interesting to show what I'm working on here, and give kind of the background of what I'm doing, as well as how we're extending that basic IPM for these multiple populations. So midcontinent snow geese breed in the Arctic and subarctic, as shown here. These different bars are the breeding locations, and the relative heights of the bars, or the heights of the bars, indicate the relative size of those colonies. They breed in the Arctic and subarctic, so Queen Maud Gulf, Southampton, and Baffin are the Arctic colonies, and La Perouse Bay and James Bay are the subarctic colonies. The birds stage from the Canadian prairies to Kansas, which is kind of shown here in the band recovery heat map. And they winter in the midcontinent of North America from Kansas to Louisiana. Now, these birds have seen some really interesting population changes over the last decades. So these are Lincoln estimates, which are estimates of population size from the band recovery data from hunters. So at one point, there were over 20 million individuals, but since 2010, this population has declined at a greater rate than it increased from the 1980s to the early 2000s. And that's really interesting. Why is that occurring? It's a huge question. Why is this occurring, and why is it occurring so rapidly? And that's something that really interests me and something that I don't know if we'll get to the heart of that with our work, but definitely it guides the questions. The objectives here for this Integrated Metapopulation Model, or IMM, are to develop this model for the 5 subpopulations over the period from 2003 until now. So we've got 19 years of data. So we want to know what demographic mechanisms are contributing to the individual subpopulation change, as well as the overall metapopulation change. And then, of course, as we are often interested in understanding what affects these, we want to evaluate different environmental drivers of demography within and among those different subpopulations. So the value of this really is to improve our understanding of the Midcontinent snow goose demography, as well as metapopulation dynamics in this huge population. This model is by far the most complicated one that I have worked with. And we have a little bit different data going into it than what I've worked with previously. So for the fecundity portion of the model, I'm working with age ratios at banding. So these birds are captured during the breeding period and banded. Usually, in most cases, the banders are targeting brood flocks, which are adults with their young. And the number of adults after hatch years and hatch years comprise this age ratio at this banding time, which can inform fecundity. The kind of meat of this model that helps us estimate kind of the metapopulation aspect of this is the banding and recovery data, which is analyzed in a multi-state Burnham model. And that is the Burnham model is essentially a survival model that uses banding and recovery data to estimate survival. And you can also, the multi-state aspect allows us to estimate movement rates among these different populations, to look at, to actually formally estimate immigration into these different subpopulations. So we don't have to rely on data or no data to estimate immigration. We can actually look at that from the movement rates. One challenge of this model is that for many of the sites, we don't actually have population size survey data, but we are using population size estimates that we obtain from a CJS model to then put those estimates into our IMM model as data to inform the population size of the subpopulations and metapopulation. I can talk more about that if you're interested in the specifics of that, but I wanted to highlight that even in this case where we have years and years of data, really amazing coverage of data, we still had some limitations that we had to overcome. And we're still working on this model a lot, but I wanted to point out that even in these optimal cases, there's some things that are suboptimal, and you have to be creative with how to get around it. So I mentioned that you could estimate movement rates. So I should have said before that this work builds on 2 very amazing previous projects. Mitch and colleagues did an IPM for the Queen Maud Gulf population. So there is an IPM that exists for one of the single populations. And then Ray Alasauskas completed a multi-state Burnham model, so not within the IPM framework, but just the survival model component to look at these different movement rates among these colony sites, which is where this figure comes from up here, this panel in the top corner. These are extraordinarily preliminary movement rates. These are some are converged and some are not converged, to be honest. And but what I want to show is that, at least in some cases, it looks like our results are lining up with what was previously done. So this is a nice check. It's nice to have work to build off of, to check yourself, to make sure that, okay, this doesn't look crazy. This looks like what we expect to see. This colony here, Baffin, is growing based on the survey data. This colony is expanding, and from our movement rates and from Ray's previous work, we are indeed showing that there is a lot of immigration, a lot of movement to that colony, and very little movement away. 99% of individuals stay here. There is a high site fidelity to each of these places, but if you were hatched here, you're going to come back. You're not leaving this place, or if you were banded there, you aren't leaving that place. So again, just like any other IPM, we're getting population size estimates, survival, in this case, recovery rates because of the Burnham model, and fecundity. I'm not showing those results today, but as in the other models that I've shown, we're incorporating the environmental drivers of those rates on survival and fecundity to understand how different environmental variation changes those rates. But I wanted to highlight the movement rates for today's talk. So this is very much work in progress. Right now, I'm at these two stages where I'm working on tweaking the metapopulation model to get it to improve estimation of those different movement rates and model convergence. And the next kind of stage and where I will hope to go very soon is incorporating the environmental drivers on those different demographic rates and look at how each of these different rates is contributing to population change at both the subpopulation and metapopulation levels. And then the last kind of big kahuna step is to combine my work with Auntie's work, looking at the Western Arctic or Pacific flyway birds and the Midcontinent flyway birds in a single model to look at movement among these populations as well as movement within. So we would get this full continental understanding of this population, or these populations, because they are pretty distinct, I guess. So bringing things back to the very beginning, where I talked about utilizing data. The question is kind of how do we best utilize data to assess species trends and demographic rates to inform management? And I presented this case where you have incomplete data sets, and this IPM is a way to leverage all of that information. I didn't necessarily speak about gaps in the data that I have here, but that is the case. These were, in some cases, incomplete and sparse data sets. And this is a really powerful approach to be able to say some important, or address some important questions and give insights into how we could more effectively manage these populations. So in conclusion, so the IPMs, I hope that you glean that these are useful for evaluating demographic rates of all species, but especially in this data limitation case. And we can use it to assess various management activities. In some cases, this can allow assessment of species that wouldn't be possible with traditional methods due to that the information that is shared across IPMs when these parameters are linked. And there's a lot of flexibility with this. I hope I've shown that today, that you can address a number of different types of questions. If you're interested in demography and how different things affect demography, there's a lot of flexibility to be creative with this to address different kinds of large-scale ecological questions. And with that, I have these were numerous projects with many funding partners. And I didn't collect data for any of this. All of this was data collected by other people, field technicians, volunteers, and I, really want to highlight that I hugely benefit from these long-term projects and other people who are willing to go out in the field and give me fun data sets to work with. So with that, I can take questions if you have time.
Thanks, Kayla. We have about 5 minutes for questions, so feel free to grill Kayla on her presentation, and then we'll do the drawing and wrap up for the semester. Let's stop this.
Oh, yeah.
I have a maybe weird question.